All right, good. It's a pleasure speaking for you. I'm Thomas. I'm a PhD student in the Francois Research Group at McGill University. And I'm going to speak about untangling a hairball, meaning a hairball of a biological network, of a protein interaction network. And currently, with the current data or methods to obtain data, we can have huge amounts of data, and subsequently we can find big models to fit this data. Now, these models do not necessarily make a lot of sense. So what we want to find what is exactly inside those models to have some mechanistic understanding of the actual process, of the actual protein interaction network. So we're going to study immune detection models, and I'll start with the adaptive immune response. So we see a dendritic cell, that's the green thing right there, which has captured some infected cell or a non-infected cell. And it will take the residues down there and present it to the T cell. Now in this receptor, the T cell receptor binds to this ligand or this antigen, we call it. And the T cell has to decide whether the antigen is of an infectious origin or whether it is from a self cell. You could imagine there's a bunch of those antigens floating around in the bloodstream. And the T cell should not respond to any of your self cells. Okay, I forgot to say one thing. If you have questions, please ask. I'd love to be interrupted. I can imagine you do this. This is a bit of a different scope than from the gene talk that we just had. So, good. We're interested in how this detection process is working. So there's a bunch of machinery in there, and how exactly this is done is not so clear. So we'll get to there. We'll first take a look at how complicated it is. Oh no, we'll first take a look at the data that we have. So people would measure the amount of immune cells, or the percentage of immune cells that respond to specific ligands. Here are the red ligands. They are qualitatively different from the green and the blue ones in the sense that they are from a foreign, or from a not self origin, and these the T cells should respond to. And we see that if they are with a sufficient number, if they are with more than 10, then the majority of the T cells will indeed be activated and go find the area of the infected area. Now those self cells for these self ligands, even if they're with huge numbers of tens of thousands, the amount of T cells that they trigger is well, minimal. So that's good. That's what we want. And this qualitative difference between some straight line here and then a sudden response and then a straight line up there above some threshold is what we're interested in. So inside the cell, it looks as follows. We have this ligand, which is to be determined self or not self, that binds to the T cell receptor, after which there's a big T cell, is going to be phosphorylated by some enzymes in there. Among others, we have SHIP1, we have LCK, and some more that are not mentioned here. So you can imagine that's a large network of arrows pointing everywhere, and interactions among them. So the moment this T cell receptor has been phosphorylated sufficient number of times, an enzyme downstream will be activated, this being ERK, which will then enter the cell nucleus and change the gene expression. So effectively, your T cell will start to proliferate and migrate to the infected area. That is the case when this peptide is from not self origin. If it's a self origin, the time of binding between this between the T cell receptor and the ligand is too short, and that means the chain doesn't have enough time to be phosphorylated multiple times. So we won't see a response. It's generally that. And then it is from the enzyme effects, mostly the SHIP1, that we have the regulatory effects. So my supervisor has published a simple model, that is this one, that it contains only 5 equations, in contrast to this one, which may have hundreds of equations. So these 5 equations, we have just the single SHIP1 molecule that summarizes all the enzymatic activity inside the cell, and we'll show, we'll have output curves that look like these. So let's see where these come from. We first have on the x-axis the number of ligands per cell. So that is the L that comes in there. They bind to the receptor. And these complexes, ligand-receptor complexes, undergo some phosphorylations until this final complex with sufficient number of phosphorylations, being about 10, triggers a response. But in the model, it's 5, it's sufficient. So here we measure the concentration of the final complex. And the moment this concentration goes above a certain threshold, you'll find the T cell being activated. And this is a little different. The model shows how a single T cell is activated, while what we measure is how a population of T cells is activated. Because of heterogeneity, your activation threshold will differ by a bit. And there's generally heterogeneous gene expression among cells. So we see here that the ligands that have a binding time of 10 seconds or more are triggered already when they are with only 10 of them, while the self-ligands that bind for a second, for 3 seconds or less, those are there, they'll never get above this activation threshold. And we know that it's due to the enzymatic activity inside the cell that we find such a characteristic curve. Because if in B, if we would remove the SHIP1 activation, we'll find straight lines, linear in the ligand concentration. So this means if there's no SHIP1 to help organizing the phosphorylation steps, there's only a very small discrimination window. If you would have like 1,000 of these self-ligands, they could potentially trigger the cell, and that is devastating. You can't have this. So we can now make a distinction between a good immune network and a bad immune network. That is, this one does well. It has this large discrimination window, and this one does not so well. And in our journey to extract the information, like the crucial components from these big networks, we'll need to make a distinction between the models that perform well, that are these, and those that do not perform well. So we have a quantification for this, which is the mutual information, which hand-wavy measures the flatness of these curves. And less hand-wavy, we can say that the mutual information is the amount of information we gain on the one variable, the binding time tau, when we measure the output variable, the output concentration. So if the output concentration is high enough, if it is above some threshold, then we have uniquely defined tau, because for whichever ligand concentration, we will also find the same output. And the way we compute the mutual information is by comparing probability distributions that we gain from these output curves, meaning we take at some steps here ligands, we compute the output, we draw them in the histogram, and then we compute this log of some joint probability distribution over the conditional probability distribution. And when there is few overlap, as here, so we have our probability here is to find an output that's always above some threshold, then the mutual information is high. Otherwise, it's not. Yes, please.
And tau 1 and tau 2 are the 2 different taus that you need for either activation or not activation?
That's right. Okay. So the tau 2 here corresponds to 10 seconds or above. The critical tau C will be below, meaning something that doesn't trigger, and be above.
Right. So you want them to have no overlap because you want one of them to trigger the immune response and one not to trigger the immune response.
exactly. And then independent of like the total number of ligands that is presented. Because you'd have a lot of self ligands that are around, but possibly only 5 of those infectious coming from a single cell that is well, infected by some bacteria, or that is sick, that has a virus inside. So then now we can quantify which immune models perform well and which don't. We take a step back. We take a look at the hairballs in immune detection. So here's our surprise. This is what the real network inside an immune cell looks like. Every node here is not even a protein, because they could summarize some more of those. So here are 700 parameters and hundreds of equations. And with this, we cannot see anything. So what we want to do, so then here are the kind of equations that we'll be looking at. So we would perform fitness-based asymptotic parameter reduction, meaning we're going to remove interactions from the network one by one, and keep the network performance or slightly enhance it. So we'll be computing for every model, every set of equations that we have, how much by changing a parameter the network performance changes. So in a flowchart, our method, phi bar, looks as follows. We have this biological network of rate equations and a fitness function, being the mutual information. For each of the parameters here, well, there will be in the models we're looking at, there will be 100 of them. We would compute the model for a parameter being an order of magnitude smaller than its initial value, and then comparing this with the initial mutual information that we had. Then we also do multiply this parameter by 10. So here some alpha would be multiplied by 10 for each of the occurrences in alpha in a set of equations. We would then solve the set of equations, find the mutual information, and test how much it differs. And for most of them, there will be a large difference. So those are all the white squares. But then here for the red squares, meaning alpha becomes much smaller and an order of magnitude smaller, and beta becomes an order of magnitude higher, we find effectively the same model. So what this means that alpha and beta always occur in the same spot in the equations, and that that only their combination is something that matters, not they're separate. So one of them is redundant. And then we check if dividing by 10 and multiplying by 10 in the limit, if that works. So we have a way of taking parameters to infinity, meaning we neglect others in the same equation term. And we check if still the fitness is the same or better than before. And if so, we remove the parameter and we continue this process. So incrementally, we would remove interactions from the model one by one until what we're left with is something super compressed and compact that is actually insightful and has captured the network performance, so the way our immune system discriminates between ligands. Now, I'm going to give an example of how we've done this. So this is one of the models. And again, new nomenclature, but about the same thing. We have some input ligands, being P, the PMHC, peptide major stability complex ligands, binding to a T-cell receptor. And this T-cell receptor gets phosphorylated. These are the Ys that stand there a number of times. And this one is going to determine, is what the cell measures, namely the output concentration. So we see a familiar face being SHIP1 here. And we see that there's also another feedback mechanism present here. And here's a ERK, that then is going to change the gene of the gene expression of the T cell if it's above or below a certain threshold. Now, for the ligands, in the output curves, we see that there's a sudden response after about 10 ligands for those with a binding time above, well, at 20, with 10, there would also be 1,000 already a response, but those below 10, they'll never become higher than an imaginary threshold we could draw here. the reduction looks like this. It's nice. First, all of this positive feedback thing would disappear. Then we have more of those that are apparently not important to discriminate in a network, and we'll end up with a simple system. So here is how the output curves change. And you see there is quite a big change. But what is not changing much is how well the detection network discriminates between the ligands. So this is a smooth function that's slowly going up until we find here with straight lines perfect discrimination. Then the thing we've been looking for is this reduced model on the right, where there's some nodes, these complexes that are being phosphorylated, and one feedback loop. An activation by this guy turns on ship, and ship then does something. So we call this the adaptation module, for the reason that if you have, it performs biochemical adaptation. So this means you have a certain input of some system. You trigger it such that there is a response. Then it slowly goes back to its starting point. And at perfect adaptation, the starting point is exactly the same as the initial point. So whatever the environment, how many ligands we have around, we will always have the same number. Then we have identified a second module here. This being the kinetic sensing module is how we call it. So this one stretches the three, or the lines apart in our output curves. So we see there is actually a difference between those taus. So if there's a large tau, that means this one stays connected long enough, so we have a higher output curve. And for small tau, this one would immediately fall back, and there's a lower concentration of the output. We have thus entangled our hairball of immune detection. We have distinguished two modules that are necessary to perform this function of ligand discrimination. And we've also identified this key node in the network, the SHIP1 molecule. And this is in accordance with what is previously known, namely that SHIP1 dictates network performance. So here's experimental data where people have been measuring the amount of T cells, the number of T cells that are responding at some normalized level. Compared to our normalized expression levels. Here, for this, when each of these members, so CD8 is the number of receptors that is on the surface of the T cell. ERK is this thing that goes inside the cell to change gene expression, and then there's SHIP1. So there'll be 70% is responding when each of them is the same. But if you would then change ERK by 5-fold backwards or forward, you would say that about the same number of T cells is responding. Then the same accounts for this number of receptors. But then if you change SHIP1 by only 2-fold, you see a dramatic decrease in responding cells. So really the negatively regulating effect of SHIP1 is tremendous. And this is what we've captured also in retrospect with our parameter reduction algorithm. So to summarize, we can with our models we can also compare them and classify them accordingly, because we can place modules back into the network, starting with this basic thing that does perfect discrimination, being here on the top, where you have one kinetic proofreading step, meaning one phosphorylation step, and then the SHIP1 activity. This means flat lines. But we know this is not biology. Biology has the SHIP1 that is not isolated, working on just one node, but working on many nodes, and there are many of those steps. So when we have n of those steps, we find non-monotonicity in the output curves, and that is something that we know exists. And the same for a feature is called antagonism, meaning certain self ligands can oppose the response to those non-self ligands. I can speak about that later. a bit more. But then if we place back this whole loop that contains ERK, so here we also find the bistable response, a sudden change. And also this is known to exist. So we have first of all deconvolved the big network into some small objects, and now we can place the modules back one by one. So we have gained some mechanistic understanding of what's inside these networks. And it's rather general applicable. You could use this in any protein interaction network when you can define, when you can quantify to what extent a phenotype is present. And we will be working towards an application in what we call informed experiments. So for this, we have to do a little better even. Biologists can very well do whole genome sequencing currently. And that's great because that's a whole lot of data. Then let's say some cancerous cells, they would compare the gene expression of the cancer cells into different stages, how far they advanced, and compare them to the regular cell. So they can gain differential gene expression. With some commercial or open source software, they can find out which of the pathways inside the cells are affected here, with what percentage. And then it's up to the PI to determine, all right, well, knowing that these pathways are affected in our cancer cells, which are the genes we're going to target? Which drugs are we going to use to target those genes to then help the cancer cells to slow down its migration or its proliferation? So that's based on scientific experience. And we think that with phi-bar, with our parameter reduction algorithm, we can help gain some more insight into those then complex signaling pathways that are there to help to formalize like this last step. So we will be informing the experiments. We could really take a big network of hundreds of genes and compress this to maybe only 20. So that would be quite informative, and I guess something PIs will be looking forward to much. But it's not so easy yet. And here we're just getting started. So to summarize, we have developed this method, phi-bar. It's a novel method for reduction of complex networks. And we can show insights into the implementation of diverse phenotypes in protein interaction networks. And I've shown how we've extracted modules and identified keynotes in these networks. And we could work to facilitate pathway analysis and hypothesis generation in the future. OK. Here's my supervisor who helped me a lot together with my fellow lab mate and Alan from Informed Experiments. There's some collaborators as well. That was my presentation. Thank you for your attention.
We'll take just one question, then we'll go to the break. So, any questions?
I can for myself, but I have many. I will ask maybe more after.
Yes, you can speak in the break.
But which one I will take? Okay, yeah. So the example you gave is, if I understand well. Oh no, I'll go with the other one. So you probe each component, or you look how changing the parameter of the equation or the value affect, if I'm not wrong, the mutual information.
Mm-hmm, exactly.
Does the order by which you look at the different components and remove them, if it doesn't change it, will have an impact on the final solution? Did you see some change, because it's like a greedy approach?
Yeah, that's right. That's right. Generally, it doesn't. So when we have this large matrix of all the parameters, and when we test just a single parameters or pairs of parameters by changing them, we will pick the one that changes the network performance the least. So either it keeps the mutual information constant, or it slightly increases. the mutual information. So this is where we base our decision on, and that's rather arbitrary to some extent, but also it's in our it's in the ideal of incremental changes and a slow progression towards a final thing. There's one exception though, where you would always find pairs, and that is actually here the case for alpha going to 10 and beta being multiplied by 10. Because I'm going to write on the blackboard here. This generally looks like in the equations that there is some nodes that the differential equations have something as follows. So, can you see it in the back? Well, it says x dot equals alpha times beta times x times y. So if we then divide alpha by 10 and multiply beta by 10, and then have for the rest the same equations, we see these are equal, but then this is also equal to multiplying alpha by 10 and dividing beta by 10. And here we would have to make a choice whether this thing, which we then call A being another parameter, alpha multiplied by beta, whether in the subsequent reductions, A is going to take place in this matrix of alpha or of beta. And that means at every reduction step, there will be one row that is removed because that parameter doesn't occur in equations any longer, either by something going to 0, so it's not there, it's just 0, or in this case where there's this multiplication of alpha and beta. And then these two rows here will be taken up by A, but it's either alpha or beta that would take its place. And now alpha and beta can occur somewhere else in different equations too, when there would be a z that is added to by beta by y again, and it disappears with some rate delta. So then, if oh, I didn't do it entirely correct. So here it wouldn't matter because it's just going to be that this A captures the exact same dependence on beta. Because if you now change A in the next step, so this is step 1, then there's step 2, then we could change z dot. We would have A multiplied by 10. So that would give these equations where z equals 10 beta y minus delta z. So that's fine. Different would it be when it's divided by 10. So then we would have that 10 alpha and 10 beta would be the same set of equations. So that the new parameter is going to be alpha over beta. And